Hi, I'm Shushan Toneyan, a PhD student at Peter Ku's lab at Cold Spring Harbor Laboratory. Deep learning has been successful at predicting epigenomic profiles from DNA sequences. Most approaches frame this task as a binary classification relying on peak colors to define functional activity. Recently, quantitative models have emerged to directly predict the experimental coverage values as a regression. As new models continue to emerge with different architectures and training configurations, a major bottleneck is forming due to the lack of ability to fairly assess the novelty of proposed models and their utility for downstream biological discovery. Here we introduce a unified evaluation framework and use it to compare various binary and quantitative models trained to predict chromatin accessibility data as an example. This framework provides easy and comprehensive model evaluation, including variant effect predictions and model interpretability for binary and quantitative modeling of epigenomic profiles. In this presentation, I'll give a brief overview of the performance, out-of-distribution generalization, and interpretability evaluation. For all example analysis results shown in this talk, we trained models to predict the attack-seq results of 15 cell lines for each input 2 kilobase long DNA sequence. First, let's have a look at performance evaluation. It is important to be able to benchmark new quantitative models against existing binary models, but the metrics used are different. Quantitative models often report correlation, while binary models report classification metrics such as AORC. Our method can evaluate models using both binary and quantitative metrics. For binary models, we take the logit values before the final output sigmoid activation. This value is generally considered to represent strength of signal detected in sequence. Each logit then represents the predicted strength of signal for 2 kb input region. We then assess the performances compared with the mean experimental coverage in the same 2K sequence and can calculate Pearson's correlation, which is a metric often used to report performance of quantitative models. And for quantitative models, we first calculate the coverage value in the predictions, then separate them into 2 distributions, peak and non-peak, according to ground truth peak calling, and finally calculate binary metrics like AORC and AUPR with these 2 distributions. This allows us to directly compare binary and quantitative models. In our analysis, we show that given our ATAC-seq prediction task, quantitative models tend to outperform binary models in both metrics. Here we're showing the binary metric evaluation performance. This is enhanced when our evaluation is done on whole chromosome, the bottom panel here, with more genomic regions with low accessibility as compared to the peak regions. Secondly, we wanted to evaluate how well models generalize to predicting variant effects from out-of-distribution sequences. Since we are interested in how mutations can affect gene regulation, a major downstream application for deep learning models is scoring the functional effects of mutations. High-performing models can inform how their predictions change relative to wild type when queried with a new mutated sequence. However, this is only informative if the models are capable to generalize to out-of-distribution sequences, in this case mutant sequences. As the ground truth for evaluation, MPRAs or massively parallel reporter assays provide a great validation dataset to evaluating how well this difference of alternative versus reference sequences is corresponding to actual biological data. Here we use the KG5 dataset, a publicly available dataset which assesses the effect of all possible point mutations on 15 promoters regions with saturation mutagenesis MPRA. The effect of each variant singularly was measured with a reporter gene, and gene expression difference was recorded. Please note that our model that we used to perform this evaluation, or all of the models that we used, were not trained on MPRA data, and they were never supplied with these new mutated sequences during training. This is a post-hoc evaluation. This is directly transferring it on a new set of related experimental data, and here we have a demonstration of an example output from a model. Visually, the signals align with the ground truth heat map on the bottom here. The model seems capable to identify important locations for function in the sequence. Also, by measuring Pearson R correlation between the ground truth and predicted variant effect, we can benchmark different models on how useful they might be for functional variant calling evaluation. Lastly, we assess the interpretability of models using global importance analysis, or GIA. While attribution maps can help to identify and footprint putative motifs, they cannot quantify the importance of motifs beyond individual nucleotides and beyond individual sequences. GIA is an interpretability approach that enables direct hypothesis testing proposed from attribution analysis. GIA computes the effect size or global importance score of putative patterns that are embedded within a population of background sequences. In our analysis, we have used dinucleotide shuffled sequences from the test set, which essentially randomizes all the positions other than the motif of interest. This approach essentially marginalizes out the confounding patterns, revealing the global importance of only the embedded pattern of interest. Here we show an example comparison of two models. The top panel shows the effects for a quantitative model, and the bottom one is the corresponding binary model. We see that both of the models identified AP1 motif, highlighted here, as important for the model to make high signal prediction, and motif lengths and distances between pairs of motifs, so when we insert two motifs instead of just one, have similar predictive effects across the two models. However, crucially, models show different sensitivity to motif interactions. The model above, a quantitative model, identified two AP1 motifs in the same sequence as more important than twofold of a single AP1 motif. This would be expected if the motif effects were additive. This suggests that this model has learned interaction between motifs while the one below, the binary one, did not. Therefore, GIA can be used as a method to identify as well as compare motif syntax learned by different deep learning models.